So, again, today, we're going to talk about replication. And I want you to understand first and foremost that replication is perpendicular, if I may say so, to the gene expression. The whole purpose of DNA replication is to copy the DNA, to pass it to the next cell, the cell of the next generation. Okay? So conceptually, DNA replication is pretty straightforward. So you have a double helix, which is being unzipped. And then each strand is copied in both directions. So the place where the DNA strand is being unzipped is called an origin of replication. The origin of replication is always AT reach. There are a lot of AT pairs. Can anyone suggest why there are a lot of AT pairs? At the origin of replication, where you have to unzip two strands. Yes, exactly. AT pair has only two hydrogen bonds, so it's easier to separate them. So, bacterial chromosomes have, for our intents and purposes, one origin of replication. Remember, those chromosomes are circular. So, one origin, and it initiates two what's called replication forks. So in this illustration on the bottom, what I just circled are two replication forks. Eukaryotic chromosomes that are linear, they're usually much bigger. These chromosomes have to have multiple origins of replication, which makes copying of DNA highly effective. Okay? Now, when each strand of the original DNA molecule is being copied, it is copied based on the principle of complementarity. If there is an A in the original strand, the newly synthesized strand will have T across of that A. There is a G, you have C, and you know the rest. Now, when the structure of DNA was established in 1953. Okay? This is when Watson, Crick, and Wilkins published a seminal paper in Nature on the structure of the DNA, suggesting that it's a double helix. Now, then the question arose, how is DNA replicated? And two American scientists, which you can say based on the last names, Messelson and Stahl, decided to figure out how it works. And they suggested three models. I'm going to present you with two. One model was conservative replication, when you have a double-stranded DNA molecule, and after copying, the original two strands remain in one molecule, and two newly synthesized strands remain in another. And alternatively, and I'm not going to talk about, dispersion replication, which is a little bit weird. Alternative was a semi-conservative model in which each strand is copied independently, so that every new double-stranded DNA contains one original strand and one strand that was synthesized from scratch. Long story short, Messelson and Stahl, they proposed the hypothesis, did the experiment, and purely based on the results of the experiment, they demonstrated that the DNA replication is semi-conservative, that each strand is copied independently, and the resulting double-stranded DNA molecule contains one quote-unquote old and one quote-unquote new DNA strand. Does that make sense, everybody? So this experiment is often considered to be the most beautiful experiment in the history of molecular biology. And if you want to, hear about that a little bit more, during the break, I can provide you with the link to the podcast where this experiment is discussed. It's a fantastic podcast, and I can give you a link to the original paper, which is available. Okay. So now, can anyone tell me what is the purpose of having two strands? Like, what is one of the evolutionary advantages of having two complementary strands?
You have more information that can be changed.
I would argue against that, because the information that is stored in each strand is essentially the same. They are complementary copies. Why would you have a copy? Just say. So again? Yes, you have a backup. You have a backup. That's the advantage. You can, if something happens to one strand, another strand, if undamaged, can be used to restore that information. Okay? Now, I want to emphasize something that I already mentioned when we discussed herpes viruses, is that the DNA replication happens strictly in the nucleus. And in general, in eukaryotic cells, and I can't emphasize it enough, eukaryotic cells, presence of the DNA in the cytoplasm, not in the nucleus, is abnormal, and there are molecular systems in place that will detect presence of DNA, whether it is double-stranded or single-stranded, and they will trigger programmed cell death. Okay? Apoptosis. So now let's take a look at the process of DNA replication. So before we dive into this image, I want to remind all of you that things that we talk about right now are processes that occur over time. Do you agree with me? What we have on our screens is the still photo. It's like you photograph a runner. You know that the person is running, but you don't know where they were running from or to. Yes? Make sense? So we will have to sort of extrapolate in terms of what we saw before and what we see now. So let's start with this enzyme right here, which is called a gyrase or topoisomerase. So a disclaimer, we are focusing on bacterial replication. Conceptually, the replication in eukaryotic cells does not differ much. Like, it's the same conceptually. There may be more proteins and stuff. So, first of all is gyrase. So gyrase is the enzyme that prevents DNA from supercoiling. Now, I know there are a lot of potentially new words in this statement. So hear me out. DNA is a spiral. In bacteria, the spiral forms additional 3D structure, which we call supercoiling. If you think about the size of DNA and the size of a bacterial cell, it has to be folded, right? Because otherwise, if it would be loose, it simply wouldn't fit into the cell. But when DNA is supercoiled, you can't actually read the information encoded in there. So the goal of gyrase is to unsupercoil, to prevent supercoiling. Okay? Once the DNA molecule, so to say, is stretched out, another enzyme called DNA helicase separates the strands. You can see the old strands being separated. Well, the problem is that these strands want to come back together, which you don't really want, because if they come back together, you can't read the information. So the protein, the cluster of proteins called single-stranded binding protein, they bind to individual strands of DNA and prevent them from collapsing back together. So now the DNA molecule is the molecules, the strands are separated, right? And you can start synthesis, or can you? So let's go back to this. So this is our one of our main players, DNA polymerase 3. So this enzyme specifically synthesizes a new DNA strand, right? That's its function. That's it. Unfortunately, this enzyme cannot just bind to the origin of replication and start copying. There are very irrelevant and somewhat complicated biochemical reasons why it's so. So effectively, that DNA polymerase 3 requires some kind of a primer. Okay? So this primer is shown in this image, as well as in these images. There's a green band. So this green band is the RNA primer. Okay? That is synthesized by an enzyme called primase. Question? Not really? Okay, so I want to ask you one simple thing. Which molecule are we trying to synthesize, DNA or RNA? Dude. No. Overall, in DNA replication, what are we making? DNA replication. It's not transcription. We're making a copy of our cookbook, right? We're making DNA. Keep in mind that we need an RNA primer to start the DNA synthesis. Are we good so far? Okay. So let's say we got this primer, this enzyme here, DNA polymerase 3, sat on that primer and started reading the original strand, the light blue one, and making the new strand the dark red. I mentioned that last week. Every enzyme that reads DNA, in which direction does it do it? 3 to 5. 3 to 5. 3 prime to 5 prime. Which you can see right here. So this is the 3 prime end of the original strand. And this is the 5 prime end. So this enzyme this enzyme right here is moving from right to left. Does that make sense to you all? Okay? So this enzyme is moving that way. Perfect, right? Clear? So it's moving and synthesizes this new strand in 5' to 3' fashion. This is a 0 out of 10 difficulty question. Look at the image and tell me, are there any obstacles that DNA polymerase 3 faces on its way from right to left? No, they're not. So the synthesis of that strand goes on uninterrupted. It's continuous, the synthesis, I mean. And this is why we call this strand a leading strand. Okay? Now, obviously, it would be a waste of resources if we wouldn't try to copy the second strand of DNA. Do you agree with me? Yeah? Like, it would be much more efficient if we also copy a second strand, right? Can I just pop the DNA polymerase 3 on the right-hand side and just start checking along to the left as well? No? Why can't I? It's the wrong direction, 5 to 3, right? So we have to kind of get around that. So the way the cell does it, and it's oversimplified explanation, but it works. So the cell employs the primase to make a primer on that specific strand. And then DNA polymerase 3, shown here, it continues it makes a short fragment, and then jumps back onto another primer, okay? And makes another short fragment, and then it's going to jump back on another primer and make another fragment. Does that make sense? Based on your facial expressions, it doesn't. So as always, and I will be totally content if by the end of the semester you will decide to drown me in Cuyahoga River for all my analogies, but I have an analogy for you. So you read a book. You read chapter 1, then chapter 2, then chapter 3. Right? Let's say the book has, 20 chapters. Now imagine what you do is you go to chapter 20, read it, jump back to chapter 19, read it, jump back to chapter 18, read it, and go through all the chapters backwards. Backwards not in the way that you're reading every word backwards. You're just reading chapters in the opposite order. Does it make sense? It doesn't? No? Okay. Now, I apologize ahead of time. We're going to have a gap in the lecture. Like right now, sound-wise, it's going to be just mumbled. I don't want to put it on pause because it may mess up the video recording. But can you help me? Since it doesn't make sense. No, I'm writing it. It doesn't. Okay. So, okay, sorry. Don't ever be embarrassed. Like if you don't understand, it's fine. So my point is that book reading is not a great example because you pretty much know what's going to happen at the end, at the very beginning, right? But you understand the gist here. So instead of working in the uninterrupted way, okay, instead of going in uninterrupted way from 3' to 5' on the proper strand, DNA polymerase, okay, I'm going to demonstrate. This is what it does. So, jumps back, synthesizes, jumps back, synthesizes, jumps back, synthesizes. Okay? I actually figured out, think of it this way. So it synthesizes forward, jumps back, synthesizes forward, jumps back, synthesizes forward. Does it make more sense to everybody? So at the end of the day, you have another strand copied right here. For God's sakes.
When it jumps back, there's no interruption?
Meaning?
Like, nothing is blocking it, it's a continuous, when it jumps
What do you mean by nothing is blocking it?
as in continuous. Like the
It keeps going. It keeps jumping. But it has to make these short fragments. Does that make sense or not? Correct. No, then it's a DNA polymerase 3 that is jumping. I'm going to be with you in a second. RNA primase just makes the primers before every jump. Because if this enzyme jumps back and there is no primer, it can't start. It needs a little bit, there's a biochemical reason for it. I don't want to get into the biochemical reason because it won't make any reasonable sense. Okay? But it has to have a small RNA bit to start on something. So primase basically prepares a starting point, one after another. And then DNA polymerase goes back to that starting point, makes a short fragment. Once it completes the fragment, it's going to jump back.
No, I think you answered it. Because as it unzips, the primase goes forward.
The primase goes here. First it started here, then here, then here, then here. It just starts at certain points on that other strand to provide a start for DNA polymerase 3.
And it goes until it runs into.
Uh-huh.
So it doesn't miss anything.
Nope.
It just
Absolutely. So now, and thank you for bringing that up, it doesn't miss anything, but it copies that entire strand in the form of short fragments. Right? They don't miss anything, they're just fragments. They're not connected yet. So these fragments are called Okazaki fragments. Obviously named after, who would guess, a dude, Okazaki, who discovered them. Okay? Now, here's a very simple question, like seriously simple question. This strand right here, the leading strand, any problems with it, any fix? No, it's all good, right? It's all good. But we need to get a lot of sort of repair on the lagging strand, right? So first of all, remember we tried to synthesize DNA. The primers are RNA. So you've got to replace them. Replace them, there's an enzyme called DNA polymerase 1. If you wonder what determined the numbering of DNA polymerases, the order they were discovered. Okay, so DNA polymerase 1 was discovered first. That's it. So this DNA polymerase 1 replaces the primer with DNA. I think of the RNA as a draft, and you're rewriting it. Okay? Does that make sense? That's the best analogy I can provide. Just replaces it. That doesn't solve the problem completely, though, because individual Okazaki fragments are not chemically linked. There is no chemical bond where I just placed an arrow. So in order to establish that chemical bond between two Okazaki fragments, you need an enzyme called DNA ligase. Ligase simply establishes the bond. So at the end of the day, the second strand is also copied, but this copying happens in a discontinuous manner. Again, look, this is like, my analogies are wrong, but some of them are useful. Okay? Reading a book from the last chapter to the first, you will still read the entire book, just in the wrong order. You will still consume the same information. Does that make sense? So this is what discontinuous synthesis. This is what happens on the lagging strand. Okay? Now, what about this table? This, I'm not even going to go over this. This is the enzyme in the left column. Its function in the right column. I will ask you questions in which you will have to match the enzyme and its function. So it can be, what is the function of the enzyme helicase? Or which of the following enzymes removes primers and replaces them with DNA? Or such and such drug inhibits DNA polymerase 3. What can you expect? And we think, okay, DNA polymerase 3 makes the new strand of DNA, so I can expect new strand of DNA not being made at all. Are we good about that? So this brings us to the very fascinating concept of mutations. Some of you may have heard the term mutant protein. It is a completely wrong term. Proteins cannot be mutant. They can be altered. Mutations are specifically changes that are introduced in either DNA or RNA. Are we good so far? About that idea, right? So, as we just discussed, the process of DNA synthesis in a cell is called what? What did we just talk about? Huh? Replication. So if you have mistakes during replication, you introduce mistakes in the DNA. You actually make mistakes in the cookbook. Okay? Like you accidentally replace, during copying, you replace 2 tablespoons of sugar by 20. That recipe is probably going to suck. Right? So DNA mistakes are very detrimental. Now, what do we call the process of RNA synthesis when you use DNA as a template? Say again? Transcription. Come on, don't be shy. Transcription. Believe me, your voice is not going to get on that recording because it's way too far. I can hear it, but the microphone cannot. Transcription, right. If you make a mistake in your copy of the recipe, is that a big deal? No, it's not. Look, okay, you wrote down the recipe, went in the kitchen, cooked it, it sucked. Because you made a mistake. You go back to the cookbook, make another copy, this time it's correct, go, the meal is perfect. So it's not a huge deal. Guess what? Well, I'm actually going to ask this question. Which process do you think is more protected from errors? Synthesis of DNA or synthesis of RNA? Exactly. DNA synthesis is very error-proof. The estimate, 1 error per 9 billion nucleotides, which is a lot. Like, seriously, the genome of E. coli is 5 million. Okay, 20 times less than 1 billion. Okay? So, these mutations don't happen during DNA replication that often. So what causes the mutations? Mostly external factors. And if you ever saw a proper sci-fi or horror or both movie, you know that the 2 things that make mutants are radiation and chemicals. So those are most common external mutagens. Now, the act of those mutagens can result in 2 very different types of mutations. The point mutations and the frame shift mutations. So let's discuss the point mutations. What you need to understand about those is, like, I give you a definition, you tell me what mutation this is. So first example is right here. So you have this triplet, GTA, the codon, right? Hear me out. If you struggle with seeing this, just tell me to make it larger, okay? So you have GTA, this codon, which encodes the amino acid valine, right? So due to the mutation, that A is changed into the T, so you have GTT now. Can you see that? No? Yes. I can make it bigger. That's as big as I can do. And I can use highlighter instead of... Okay. So do you see this codon right here, GTA, that I just highlighted? It encodes valine. Mutation changes the last A of that codon into T. Yeah? That codon, GTT, still encodes valine. There's no change in amino acid. So protein remains the same. We good? We call this mutation silent, because this mutation does not result in any phenotypical change. As you can imagine, the reality is more complex than what I just said. But again, right now, mutation that does not alter the sequence of amino acids is silent. What if you have this gene, CCC, which encodes proline, but for some reason the first C of that codon is changed to ACC. Can you see that? So that's a mutation, right? That point mutation, the replacement of C with A, results in a codon for a different amino acid, threonine. Can you acknowledge that? So I can tell you that proline is hydrophobic amino acid and threonine is hydrophilic. Do you think that replacement can alter the structure of a protein? Yeah. This altered structure, will it alter the function? Yeah, very much so. Yeah, it will. Well, I want you to think about it as not yes or no answer. I want you to think about it as a spectrum. In some cases, if you replace one amino acid by another as a result of a mutation, amino acids can be very similar in their properties. Do you follow my idea here? So although it's a different amino acid, it may not have a huge effect on the protein. So even a missense mutation, when an amino acid changes, it may result in a different structure and function, or it may not. Okay? Does that make sense? Okay, good? Sure, go ahead. G? Like instead of A, you would have G like guanine? You mean, hold on a second, I just want to make sure that we're talking about the same thing. Instead of A here, there would be G. Yeah, let's take a look. I don't have it here. If you have a phone nearby, you can Google what GCC codon encodes for. And I don't really know, and my phone is far. No. It's actually a good question. Like, what can it be different? I don't remember. I believe it's alanine. Okay. You asked a very good question. Proline is a pretty bulky non-polar hydrophobic amino acid. Alanine, GCC, isn't as bulky but still hydrophobic amino acid. Will it change the structure? It will. Will it be as detrimental as replacing a hydrophobic proline with a hydrophilic threonine? Probably not. So there is a lot of nuance to mutations. Any questions about point mutations? So understand that it's a replacement and be able to match the description with one of three types. Now, what about the frameship? Oh, hold on. Nonsense mutations. Completely forgot about that. So, right here, you can see a codon, TAC, in the DNA, which encodes an amino acid tyrosine. If you change it to TAG, instead of a tyrosine, it's going to be a stop codon. Translation is going to stop. Does that make sense? So that will cut the protein short. So these nonsense mutations are much more detrimental than any other type. I mean, it's going to make a protein. Okay, you're reading a book that normally consists of 20 chapters, but your version ends in the middle of chapter 5. This book sucks, right? This protein sucks. Now, hear me out. If your book stops at like few words before the end of chapter 20, it's fine, right? Like you pretty much have an idea. So it depends where this nonsense mutation is. If it's at the very end, it may not be super detrimental. If this mutation is in the beginning, that has much more effect. Now, what about frameshift mutations? So when you look at the process in which ribosomes read the genetic code in RNA, they read it as frames of 3 nucleotides that we name codons. Can you see the codons that I highlighted with red brackets? Yeah? So now imagine you remove these 2 nucleotides from the original DNA molecule, so that entire chunk shifts to the left. Now, first triplet codon is the same. But then, the next one is different because TA are gone and replaced with CC from the right. And the next one is different, and the next one is different. So you have a massive change to the amino acid sequence of the protein that's encoded by that mutant gene. No, they can happen to an RNA, but the thing is, mutations in the RNA, if you talk about a cell, not a virus, mutations in the RNA aren't passed down to the next generation. The worst that can happen as a result of the RNA mutation is death. But it will not be incorporated in the original cookbook. When you make a copy of a cookbook, your cookbook is not altered. When you make a copy to cook meals in the kitchen, like when you scribble down the recipe, right? But if you're actually copying the entire cookbook by hand, and you make a mistake and you write down 20 tablespoons of sugar, this recipe is going to suck all the way down the line. Does that make sense? Pretty much, yeah. The entire gene is altered. It's not just one amino acid, it's multiple amino acids. Go ahead.
Can it only be two little genes, or does it have to be?
What do you mean, two nucleotides? No. Can it be one? Now, what if 3? Are we going to have the shift of that reading frame? No, we're just going to lose an amino acid. Does that make sense? So if this is the number with a factor of 3, 6, 9, whatever, we're going to lose a certain number of amino acids. But overall, the rest of the sequence will be fine. If it's 1, 2, 4, 5, 7, 8, something that is not divisible by 3, then we're going to have a really screwed up protein. Did I answer your question? Okay, awesome. So let's talk about some of the mutagens. And many of them actually carry a lot of clinical importance. So one type of mutagens that I want to highlight is the nucleotide modifying agent. An example of this would be nitrous acid. So it converts the GC pair. Okay, so GC pair becomes converted into AT. You don't need, I'm never going to ask this. The only thing I want you to understand right here is that nucleotide modifying agent changes one pair into another. Which into which, it's fine. We don't need to know that. How's that clinically relevant? So, yeah, so nucleotide modifying agents convert one pair, in this case, GC, into another, AT. Okay? So what's the clinical relevance of that? Some of you may eat cured meats. Corned beef, cured bacon. Yeah? What is the curing agent? Can you be a little bit more specific? It's not salt. It's not salt, the table salt. It's sodium nitrite. And sodium nitrite that is used for curing those meats can be converted to nitrous acid. And there's a study which was issued 2 years ago from Britain, huge logical study, showing the link between cured meats that are treated with sodium nitrite, and the risk for developing colon cancer. Which kind of makes sense, right? Because if you introduce mutations through your curing agent, you increase the risk of cancer, right? So now, if you go to a grocery store and start looking, there are a lot of uncured. They don't use sodium nitrite. Okay? Which, it begs a lot of questions that I don't want to dive into, and those questions aren't about microbiology, but more about economics. Okay? And honesty of corporations, which doesn't really exist. So another type of mutagens, chemical mutagens, are so-called intercalating agents. Now, my examples may seem pretty far-fetched. So this is acridine orange. It incorporates itself between the bases, like gets stuck in the chain. It intercalates. So you can imagine that this molecule right here will mess up the DNA structure. So when DNA is replicated, you're going to have deletions and insertions. Deletions and insertions are kind of a big deal, right? Would you agree with me? But why do we even care? So I'm going to go on the limb and suggest that you hear the names of these chemicals, acridine orange and ethidium bromide, for the first time in your life. Maybe not. I would be happy if you prove me wrong. Like, seriously happy. Those are the chemicals that bind to DNA really well, as we just discussed, and make it very visible. They are very useful in the lab. I used both of them. They are super useful. They are also extremely carcinogenic. You're not going to run into them in your day-to-day life, aren't you? You're going to run into other things. Do I have... next page. I have polycyclic hydrocarbons. Sounds very 3-hydroxy chicken wire. Posh, yes, ostentatious. People who smoke inhale those compounds on a regular basis. And it doesn't matter what you smoke. tobacco, weed, any product of grass burning will produce those polycyclic hydrocarbons that have these intercalating properties, and they will negatively affect cells in the lungs and lung cancer. Make sense so far? Now, radiation. Well, that's kind of easier. So we have ionizing and non-ionizing. So ionizing radiation includes X-rays and gamma rays. And it's pretty brutal. And it introduces either single or double strand breaks. Okay, you can imagine that you can restore a single strand break because you have another strand unaffected. Double strand breaks is a bit of a big deal. Okay. UV light, which is a non-ionizing radiation, forces adjacent pyrimidine bases, mostly thymines, to form this bonds. So we call this structure a thymine dimer. As you can imagine, forming that dimer results in a disturbance to the structure of the DNA, and when it is copied, mutations arise. Okay? So this table summarizes the agents, the mutagens. So now, let me clarify it for you for the purpose of exam, so you don't get hung up on the things that you really don't need to know. I'm not going to ask you about the names of individual chemicals. Am I making sense? Rather, I can tell you this. What if we use nucleoside analogs? What's going to happen? And tell me, point mutations are going to happen because one type of base pair is converted to another type of base pair. Now I'm going to let you lead on this answer. Do you think I can ask you, is it AT to GC or GC to AT? No. Just want to understand. Yeah, that's a point mutation that we make. Okay? Good. Nucleotide modifying agents. Same story. Point mutation because one base pair is converted into another. Intercalating agents. What do you need to know? First, how they work. Disturb the helix. What type of mutations? Frame shift, deletions, and insertions. Am I being clear so far in my expectations? Okay. Radiation, ionizing. What they are, X-rays, gamma rays. What they do. Okay. And then the types. The types, hear me out. For all types of radiation, they can cause point mutations. They can cause frame shifts. You can't really predict. Okay, so for instance, for ultraviolet radiation, what is the mechanism? Pyrimidine dimers. What type of mutations? Frame shift to point. Am I clear? Okay. And obviously, if we wouldn't have a way to pair the DNA, we'd be long dead. Like seriously, would be dead. And cells will be dead. All cells will be dead. So how do we prevent this? I mentioned that DNA replication is an accurate process. And effectively, DNA polymerase 3 has a proofreading capacity. What do I mean by that? So imagine you have a DNA sequence, ATGC, right? And you make a new strand, so you put T here, agreed? And A here. But then for some reason, DNA polymerase inserts T across the G. Is that correct? So every time DNA polymerase 3 inserts a nucleotide, it checks. Does it match? And if it doesn't, it will remove it and insert a proper one. Okay, this is called proofreading. As any other mechanism, it's not perfect, makes mistakes. So let's say, and we're going to discuss the damage to the DNA that is caused by the UV light. Let's say you expose DNA to UV light, and you have this thymine dimers formed. You all clear on that? Okay? So this DNA is obviously, this strand is obviously not well. So we need to fix that. Do you agree with my statement right now? Excellent. There are two options. If there's still plenty of light, the light will activate the enzyme called photolyase. And photolyase will do the simplest thing possible. It will break this bond. Thymines are not connected to each other anymore. You go back to square one. It's all fixed. Agreed? What if there's no light? Well, the first thing is to cut out the defective part of one of the strands. So the enzyme called endonuclease cuts right here. And then your old pal from the process of replication, the enzyme called helicase, takes the damaged piece out. So now you have this gap, right? Right? And you need to fill this gap. Another friend of yours, DNA polymerase 1, will fill the gap. And DNA ligase will make sure that it is chemically attached to the rest of the restored strand. Okay? Now, I have a question for you. When you, like today was a beautiful weather, I hope you had time to spend outside, at least maybe like a walk, but you probably went outside for a little bit today, right? Were you exposed to sunlight? Yes, you were. Did cells in your skin, for example, accumulate mutations? Most certainly. But these mutations got repaired. The problem arises when the repair systems cannot keep up with the rate of mutagenesis. Does that make sense? So like if I will stick you stark naked into the tanning bed for 18 hours a day, you're going to get skin cancer in a week. Okay? Because your repair systems will be absolutely overwhelmed. Do you understand the idea? It's a fight between the mutations and the repair mechanisms. Any questions so far? So this brings us to one of my favorite concepts in the entire study of microbiology, the Ames test. So it is used to test chemicals for their ability to cause mutations. So let me ask you a question. Let's say I have a new drug. I want to see if it causes mutations in DNA, right? Is that a good idea to give that drug to mice and see? Yeah, you think so? Where are you going to... Okay, hold on. No, it's a good idea. But let's say you give it to mice, and mice don't die. One day they don't die. 2 days, 3 days, 4 days. Can they tell you if they feel bad? No. So you think, oh, maybe they developed a tumor. Where in the mouse are you going to look for a tumor? Why not brain? So anywhere. That's a lot of mouse to dissect, right? That's like slicing it out in the very fine slices. So instead, you can test if a chemical has the potential to cause mutations. And for this, you can employ a wonderful test called an Ames test. So I can tell you that there is one question on the exam about the Ames test, just one. It is, in a way, the hardest question of the exam. Let me explain why. Hold on, I'm not trying to scare you. If you understand the Ames test, the question is easy. If you memorize it, yeah, good luck with that. It's purely the question about understanding. You can say, oh, screw that, I'm not studying Ames test. I'm just going to that question, I'm just going to guess. Fine with me. But what I'm trying to do is to explain how it works, okay? So bear with me. This is your agar plate. It contains a bunch of colonies of salmonella. Okay? The medium that you have here, the medium in that plate, contains an amino acid histidine. So cells, bacterial cells, don't have to make their own histidine. Agreed? They can just get it from the environment, right? They don't need it. Okay, we're good? So we do replica plating. It's a little velvet pad that you press against the original plate and transfer those colonies onto other plates. So this plate has histidine in it. This plate does not. Okay? Remember, any cell requires all amino acids to grow. Okay? So imagine there is a cell on this medium that can make histidine. Will it grow? Yeah. If it can make its own histidine, it will. And this is what we see. What if a cell can't make its own histidine? It's not going to grow. And suddenly we discover that there's a colony that grows in the presence of histidine but doesn't in the absence of it. Am I making sense? So the microorganisms that are present within that colony, the cells of Salmonella, they are auxotrophs. They need a specific nutrient, namely histidine, to grow. Does it make sense to you? Yeah? Okay. Now I'm going to go on an illustration spree, which you probably would not really acknowledge. Here's the question that I have for you folks. The ability or inability to make histidine. Where is it written? That's the best I can do with a lead. The fact that cell can make histidine versus the fact that the cell cannot make histidine. What determines that? Say again? The gene. Right? So imagine you have DNA. And here is, let's call it a histidine gene. If it's functional, cells can make histidine. All good? For some reason, that cells in this colony, lost the functionality of that gene. Do follow me so far in my logic. So what it means essentially is that there was some kind of a change, and now you have the DNA molecule and the histidine gene. There is some kind of a mutation inside of it. Okay, so that the dot is the mutation, which renders this gene dysfunctional. Everybody's with me on that? Now here's what you're going to do now. You're going to take your cells that require histidine right here on the right, and you're going to expose those cells to the chemical that is potentially mutagenic. Hear me out. If the chemical is mutagenic, it's going to introduce mutations into the DNA. Do you agree with that statement or not? Yes? Good. So these mutations are going to be random. Because it's just a chemical, it doesn't have any preferences to a specific location. Agreed? You have a lot of bacterial cells in your sample. And there is a very non-zero chance that the mutation will change the wrong nucleotide in the histidine gene into a correct one. In other words, two negatives will give you a positive. Okay. I think I have a sort of, let's say I am copying the English text. Hear me out. And the first word is meat. So when I copy, I make a mistake. Now it's meat. Right? It's absurd. Somebody else makes a copy. They genuinely think that this is the word that's supposed to be, but they make a mistake again, and we go back to the original. Right? This is called a divertant. Do you understand what I'm trying to say? Like, two wrongs make right. Okay? Y'all with me? If there is a diversion, this revertant, will it be able to make histidine? Yeah. This revertant, if it reverts because of mutagenesis, will it be able, since it can make its own histidine, to grow on a medium without this amino acid? Yeah. So what you do, you plate your exposed cells onto the medium without histidine. Does that make sense to you? If your chemical, possible mutagen, if it was indeed mutagenic, you're going to get a bunch of revertants, and you will see a bunch of colonies on the medium that lacks histidine. Clear? If your chemical is non-mutagenic, this reversion right here isn't going to happen. So your bacteria is still going to be deficient. Do follow my train of thought. Kinda? I'm going to go over it one more time. And they will not produce any colonies. So I'm going to lead with the insane analogy, like absolutely outrageous, just absurd, okay? So I'm a fairly healthy person, give or take. Okay. I may have some internal problems. But if you will try to test if the chemical is mutagenic on me, you don't know where to dig. Because if you use it on a healthy person, like, where are you looking? Okay? So what you do, you give me colon cancer. Okay? That's your disrupted histidine gene. Okay? My colon cancer is because of a mutation. So I fucking shit myself, and I'm in terrible shape. Okay? You give me a chemical that you want to test. This chemical introduces mutations. And it's a very good chance that this chemical will introduce a mutation that corrects my colon cancer back to normal. I told you that it's absolutely outrageous, just horrible analogy. But if I am cured of mutation-caused colon cancer, the chemical that you gave me is mutagenic because it's a mutation that corrected the original mutation. And if I'm still dying, good news for you, your chemical is fine. It's not mutagenic. Yeah. You're absolutely correct. This is why I told you that my analogy with the human is horrible. Bacteria, it will mutate. Everything all right. But we aren't looking for any other phenotype. We're testing only for the ability to synthesize histidine. We're just not looking at other mutations. We fully acknowledge that they can happen. We're focusing on that single gene. You want to know if it can correct the ability to grow without histidine, and that's it. That's it. It does not test for any other changes which may happen. Go ahead.
Okay, but even then, you said there's a non-zero chance that it will change that specific gene, like and revert to a functional one.
This gene.
Yeah, so don't you have to do it, so would you have to do it on like a giant scale?
Oh, yeah, dude. In that tube, you have probably trillions of cells. That's enormous scale. So even if you change, even if this repair happens in 100, it's like you shooting a shotgun at a genome. Okay? It's a big-ass genome. There are a lot of pellets. But since there are so many genomes, and you have an enormous number of pellets, the chances of one pellet hitting that one single specific mutation are pretty much non-zero. And like very much non-zero. If you would try to do it with 10 cells, it wouldn't work. It's not enough. Okay? So you have to have a certain number. There is a statistics. I passed statistics in college. And by passed, I mean it went by me. So don't ask me. But you can do some statistical correlations based on the probability of mutations and the length of the genome and the amount of the mutagen and how many cells to figure out the optimal conditions for testing. Okay? Does that make sense? The reason why I like this this conversation is because it illustrates the dependence of genotype, of phenotype on the genes that are or are not being expressed and how mutations can affect that. Okay? That was sort of an apex of complexity. On this, we're going to go downhill. Go ahead.
So, we want to know if it affects the histidine phenotype. Is our goal to look for the negative, say, meaning like it doesn't affect in a clinical scenario, right? Like we have a potential mutagenic whatever
Chemical.
chemical.
Yes.
And so would we want to test it so that we are able to give it to people without having to worry about it changing genetics?
Correct. So the goal of Ames test is to filter out the chemicals that obviously are mutagenic. Okay? If Ames test shows that in bacteria, this chemical does not cause any mutations, we can proceed with clinical trials. But if it alters the DNA, we probably shouldn't give it to mice or humans or whatever. Like it's already cut off. Does that make sense? So now we're going to chat about the gene transfer. I'm not going to dive into the entire Griffith experiment. It's really cool mechanistically. So basically what the discovery that Griffith made, the discovery of the process of transformation, was, I would say, a bit accidental, because Griffith was looking for a molecule that carries genetic information. He was trying to establish what molecule actually conveys genetic information. By the way, his conclusion was wrong. He thought it was a protein. But what he demonstrated effectively is that bacterial cells, you see these blue cells? These blue bacterial cells are pretty much benign. They don't kill mice. They are very much alive. And turns out these live bacterial cells can collect the DNA from the dead bacterial cells, from the environment, incorporate that DNA into their own chromosome, for example, and turn into these bad cells. Okay? Remember, these bad cells, they kill. So here's what it means. Do you see the bacterial cell right here? Do you see the DNA fragment, the plasmid? So cells can obtain that plasmid from the environment, incorporate it into their chromosome, and start expressing the genes that are present in the plasmid. Okay, whatever those genes are, those genes protect bacteria from antibiotics. After the transformation, bacteria become resistant to antibiotics. If they contain some kind of virulence factor, they can start producing that virulence factor. Now, let me ask you a question which is like 5 or 6 or maybe more out of 10. It's not really a complicated question, to be honest. Can we use transformation to our benefit? How? Didn't hear the last part. Couldn't? Oh, technically you can. But I have even a better idea. Let's say you want to make a buttload of cheap insulin. Insulin is a protein. Insulin is encoded by a gene. What if you put an insulin gene in the plasmid, stick in the bacteria, and bacteria will start making insulin? Sounds attractive. This is pretty much how companies make insulin. They just don't use bacteria, they use yeast. But similar idea. Okay, so it can be bad, but it can be used for good. Does the concept of transformation sound clear? So bacteria get DNA from the environment and acquire whatever. Okay, transduction. So for transduction, very brief introduction into the cells, into the viruses of bacteria. Viruses of bacteria are called bacteriophages. And there are 2 types. First, we're going to talk about lytic bacteriophage. You can see an illustration right here. So this is the bacteriophage. The purple color signifies the bacteriophage DNA, and the green color shows you the bacterial one. Okay, you with me? So bacteriophage gets in and chops up the DNA of bacteria. And then new particles are made, and most of them package proper phage DNA, and this guy doesn't. It packages the fragment from bacteria. Pretty much, if you have kids and they play like soccer, for example, like my kids play soccer, okay? Every time after like the game or practice, there's a slew of emails saying, "My little Johnny brought home somebody else's backpack," or, cleats or whatever. So they get the stuff mixed up all the time. Does that make sense? This is what happens here. This poor bacteriophage packaged the wrong DNA. It can still go away, get on another bacterial cell, infect it, and now the DNA from the first cell gets into another bacterial cell, gets incorporated, and gives it some kind of extra powers. Make sense? So this is called generalized transduction. Okay? It's sort of non-specific. The rate at which it happens is astonishing. Okay? And yeah, it's pretty common. Now, another type of transduction is specialized. Okay? Specialized transduction is carried out by lysogenic phages. So let's take a look at what's going on. So the image here is not great. I'll try to make the best out of it though. So this is the phage. Yeah? You see the blue DNA. So this DNA gets in the cell and gets incorporated into bacterial genome, right? So it's there. Bacteria produce and reproduce, and everything's fine. Everything's hunky-dory. Then bacteria get under a stress, and the phage DNA cuts itself out. Do you see that? The blue stuff that is being removed. Now, it's not perfect. The cutting out isn't perfect. So I have this little fragment of bacterial DNA that is accidentally attached to it. So phage makes new particles, packages the DNA. You see? And it now... Okay, so to wrap up our conversation on transduction, okay, we figured out basically two things. That there are viruses that infect bacteria, that these viruses can carry genetic information from one bacterial cell into another, and that the mechanism of that transfer differs based on whether the phage kills bacteria almost immediately, then it's a generalized transduction, or the phage is lysogenic and it incorporates itself into the bacterial genome, then it's a specialized. This is one of my favorite gene transfer mechanisms. Conjugation, sometimes microbiologists refer to it as a bacterial sex. So what happens during conjugation? This is the F plus cell, so-called donor cell. It contains an F plasmid. Okay? So in this case, F stands for fertility. Obviously, it's not really sex. So this plasmid is being transferred, one of the copies is being transferred into the recipient cell that doesn't have a fertility plasmid and therefore is designated as F minus. Am I making sense? Yeah? So after the transfer, the original donor cell remains F plus, and the recipient cell turns from F minus to F plus, since now it contains the fertility plasmid as well. Okay, so the term bacterial sex obviously refers to the transfer of genetic information from one cell to another. I want you to be very clear about that. Bacteria do not have sexual reproduction, period. Okay? This is the reason why bacteria employ these gene transfer mechanisms, because they don't have sexual reproduction. Can anyone tell me what is the biggest advantage of sexual reproduction? All this crossing over and...
You get like a bunch of new genetic information.
Excellent.
New.
Yes, you have genetic diversity. The lack of sexual reproduction means you don't have that genetic diversity, right? Horizontal gene transfer, conjugation, transduction, and transformation solves this problem. Now, if you have a question about whether or not the high-frequency recombination will be on exam, it won't be on exam. This part. Okay, I'm not going to ask anything about HFR cells. Okay? All good? Okay. So we're done with the gene transfer. Now we need to conclude this with the regulation of gene expression. So we're going to talk about riboswitches first and then get to the operons. Riboswitches, and this is what I love about or hate about molecular biology. Conceptually, it's a very simple idea. What I just circled, is that a structure within RNA? Yeah, it's a hairpin loop. It's called riboswitch. Do you think it has a function? Yeah, it does. It's a switch. When a certain specific molecule binds to a riboswitch, it changes its structure. Would you agree with that statement? Yeah. When structure changes, does function change? Yeah. So, effectively, this riboswitch and this riboswitch are in the on position. Ribo refers to the fact that those structures are present in RNA. And RNA participates in two processes. In one process, transcription, it participates as a product. In another, translation, it participates as a substrate. Both processes, transcription and translation, are regulated. One of the ways to regulate it is by a riboswitch. So, imagine there's a small molecule that binds to the switch and turns it from an on to an off position. If this specific riboswitch regulates translation, translation stops. If this specific riboswitch regulates transcription, transcription stops. Does that make sense? Yeah? It's a switch that is activated by some kind of a small molecule. That's all you need to know. And you need to understand that it regulates specifically transcription or translation found in the RNA. Am I making sense? Okay, excellent. Where do you keep your forks? At home?
In a drawer.
Where?
In the kitchen.
Kitchen. What about spoons?
Kitchen.
Same? Not in the garage? Could you keep it in the garage?
Depends on the spoon.
I mean, you could, right? There's no problem with that. It would be inconvenient to walk to a garage to get, like, forks and go to the bedroom to get the spoons and to go into the kitchen to get the butter knives, right? So what is the reason you keep all those utensils in the kitchen? That's where you need them, right? You keep them all together. So bacteria do very similar thing with genes. Genes that are related functionally to each other, genes that work on the same project, let's put it this way, are organized in operons. Okay? So this is an example of an operon. I want you to focus on that chunk of it. So A, B, C, and D are what's called structural genes. These are the genes that encode actual proteins. Does that make sense? Like all these proteins, they will work together on something. Remember the term promoter. I mentioned that. This is where RNA polymerase binds to start transcription. But turns out, in the operon, there's also an operator sequence right here, which controls whether the structural genes are going to be expressed or not. Before we move on, does it make sense? Okay. Many operons have regulatory genes. They regulate. That's it. Let's move on. I'm not going to talk about those. I'm going to focus on that box. So there are two types of operons, inducible and repressible. I'm going to give you an example of an inducible operon using lactose operon and repressible operon using an example of tryptophan operon. Are we good so far? I can promise you that the questions about operons that will be in the exam are going to refer to these two examples, period. Am I clear? So first, let's take a look at this inducible lactose operon. Okay. So these are the genes that encode proteins necessary to break down milk sugar lactose. Am I making sense? So if a cell has lactose available, these enzymes, I'm going to give them like Z, Y, and A. These enzymes break down lactose in some sort of products that can be used, like basically it's a nutrient, right? So what you see in front of you is the enzymatic reaction, and lactose, in this case, is a substrate. So far, statement makes sense? Okay. Now, let's get back to the operon thing. Do you see RNA polymerase? Yeah, it is bound to the promoter. Do you see the repressor protein? Repressor is bound to the operator. Can RNA polymerase transcribe? Why can't it transcribe? There isn't a repressor in its way, right? So when lactose becomes available, it binds to the repressor, and that binding removes the repressor from the operator. Okay? Can polymerase transcribe now? Yeah, it can. When it transcribes those genes and eventually the proteins are synthesized, those proteins, what are they going to do to lactose? What are they going to do to lactose? I just told you this. Digest it, yeah, break it down. So lactose will not exist anymore, right? And when there is no lactose, what's going to happen to the repressor? It's going to bind back to the operator. So only when lactose is available, these genes are going to be expressed. Because why would you need to express them if there is nothing to break down? Does that idea make sense to you all? So lactose in this case, which is a substrate of the enzymatic reaction, it induces gene expression. This is why we call the lactose operon inducible. Good? All right, now let's talk about tryptophan operon. Does transcription happen at the top part? Here, yeah. Okay, so this transcription results in the expression of these 5 genes. All these 5 genes are essential to make an amino acid tryptophan. So we can present it this way. Okay, so you have reactants, and I'm going to use the letters to name the proteins, okay? E, D, C, B, and A. Okay, these genes convert reactants into an amino acid tryptophan. Okay? Pretty logical, right? So you have more and more tryptophan in a cell, correct? Eventually the levels of tryptophan reach high enough levels so it starts binding to the repressor, and only when it binds to the repressor binds to the operator. Can transcription proceed now? So tryptophan, being a product of the biochemical reaction, represses its own synthesis, represses the operon. Does it make sense? Yeah? I think repressible operons are much easier to explain. It's like, you come home, there's no food in the fridge, you cook something, right? You come home, your fridge is stacked with all food that's ready to eat. You're not going to cook anything. Your cooking activity is repressed. Okay? So the presence of the product inhibits its cooking. You don't need any more tryptophan, you have plenty already. Eventually, a cell is going to use it. When it runs out of tryptophan, those genes become expressed again. Does that make sense? There are going to be a couple of questions on the operons in the exam, and these are 2 operons that I can ask you about. So now we're switching gears completely and start talking about metabolism. Right now I'm going to introduce some basic concepts, and then we're going to move on upstairs. So the term metabolism belongs to all processes, chemical processes in the cell. Metabolism can be divided into catabolism, which is degradation, and anabolism, which is synthesis. Okay, when you break down something, the catabolic reactions mostly release energy. We use the term exergonic. Anabolic reactions, reactions of synthesis, usually require energy. So if you look at the example here in the center of the screen, so glucose, its catabolism, breakdown, results in the formation of carbon dioxide and water, but also provides for anabolic reaction, the blue arrow of ATP synthesis, okay? So exergonic, catabolic, provides the energy for endergonic, anabolic. Am I making sense? No? Would you agree that to make ATP, you need to invest energy? You can get this energy by breaking down, say, glucose. when you break down things, we call it catabolism. Okay? When you break down glucose, this process releases energy. Therefore, this process is catabolic and exergonic. We good? It's like you burn wood. That energy goes into the process of ATP synthesis. Synthesis is anabolic. Does it make sense to you all? And it requires energy. Okay? ATP itself can then break down, so catabolic process, ATP breakdown is catabolic process. When it is broken down, it releases energy. This energy can feed another anabolic process. If you prefer an example from the human standpoint, not bacterial standpoint, imagine that you decide to become a bodybuilder. You're going to get juiced. There's no other way. You're going to get anabolic steroids, right? No, seriously, that's true. You won't be able to become a professional bodybuilder without steroids. I know a guy who competed. He told me that outright, okay? So you're going to get those anabolic steroids. They will stimulate the muscle growth. But to grow the muscle, you need a lot of energy. You can't grow muscle when you starve. Does that make sense? So let's say I'm incredibly fat and I decide to become a bodybuilder. So I'm going to take anabolic steroids to stimulate my muscle growth, but the energy that I'm going to get is going to come from the fat that I'm going to break down. So the breakdown releases energy, synthesis requires energy. Does it make better sense? Okay, cool. So now what about, what about this? The stable. I'm not particularly proud of it, to be honest. I'll explain why. So every living thing needs energy and carbon to grow. So energy can be derived from chemical sources or from light. Organisms or cells that derive energy from chemical sources. Those organisms, we call them chemotrophs. Organisms that derive energy from light, we call them phototrophs. Am I clear? What about the source of carbon? Source of carbon can be inorganic, like carbon dioxide, or organic, like dead or live animals and plants and whatnot, okay? If the source is inorganic, if a cell can convert inorganic carbon into organic form, we call them autotrophs. If cell has to consume organic carbon, we call them heterotrophs. And if you think about naming, it makes perfect sense. Auto means self. So self is self-feeding. Heterotrophs means that some other organism has to provide this food. So to simplify, obviously, no, I'm not going to ask you about examples like this, but I will absolutely ask you to match the characteristic of an organism and the appropriate term. So if I tell you that purple non-sulfur bacteria receive energy from light and carbon from organic compounds, you think, okay, energy from light is photo, carbon from organic compounds, hetero, photoheterotroph, and you pick the answer. Make sense? Good? Right. last thing for today. So, okay. Most of this information won't be on the exam directly, but you have to know these terms to answer questions. And these terms are oxidation and reduction. Okay? So, I'm going to try to work with this. So, oxidant gains an electron and becomes a product. Okay, reductant loses the electron and also becomes a product. But what I want to demonstrate to you is the link between these two processes. So now take a look at this. Imagine that you have a compound A. Wait a minute, what just happened? Okay. Imagine that you have a compound A. Sounds good? And you have another compound, we're going to name it B. A is losing electrons, B is gaining them. So in this scheme of things, A, since it loses electrons, is oxidized, and B, since it gains, is reduced. We good so far? So, I work for Tri-C. I'm getting paid. By what organization? Tri-C. Which organization pays me? Tri-C. I am being paid by Tri-C. Tri-C pays me. A is oxidized by B, okay? And B is reduced by A. So A, that's the analogy of the payment. A reduces B, B oxidizes A. So A is a reductant, B is an oxidant. No, we done. None of this directly will be on the exam. Let me explain. But I will be asking you questions about metabolism. And the questions will be like, in the electron transport chain, NADH is blah by the proteins of electron transport chain. Reduced, oxidized. Does that make sense? And if you will approach me and ask me, what is that? What is oxidized? I'm not telling. So what I'm doing now, I'm giving you the terminology that I'm going to be using. Okay, that it is your responsibility to understand what it means. Like if something is oxidized, what does that mean? It's going to lose electrons. If something is reduced, what does that mean? It's going to gain electrons. Okay? So that was the entire goal. Now, all energy transfers in the cell are mediated by electrons. When chemicals are processed to retrieve energy, it's all about stripping electrons. It's all about oxidation, burning effectively. When glucose is metabolized, glucose is kind of slow burn. Okay? The problem with electrons is that they cannot exist independently. There is no like a single electron just floating around the cell. they have to be carried. And for this, there are electron carriers. I call them the electron taxis. Okay? Do you need to know the formulas? No. You don't. You don't need to know the formulas. But you should be able to recognize this. So this is NAD+. Oh, crap. So this is NAD+. Okay, this molecule. It can acquire electrons and a hydrogen and become NADH. When a molecule acquires electrons, when it gains electrons, is it oxidized or reduced? So this is a reduced form. Now, the reaction can go backwards. NADH will give electrons away at some point. When a molecule loses electrons, is it oxidized or reduced? Oxidized. So NAD+ is the oxidized form. All good so far? Same story with FAD. FAD receives electrons, forms a reduced form known as FADH2. When FADH2 gives electrons to something else, it forms the oxidized form, okay, FAD. Okay? Now before and all this electron transport, the whole goal of this electron transport is to provide electrons to a system that will be able to generate ATP. Okay? Now, I use the term electron taxis for these molecules, right? So they shuffle electrons within the cell. Do you follow my train of thought? So let me ask you this. Let's say you step out of Tri-C and hail a taxi and you get in. Is your ride going to end? Hopefully, right? Hopefully they're going to drive you to your house or wherever, and you're going to leave the taxi, and the taxi is ready for another passenger. This is a critical point for these molecules. These molecules do not hold on to the electrons. The purpose of this is to get an electron, which is important, and deliver it somewhere and unload it, which is equally important. So when next week we're going to talk about metabolism, I want you to keep in mind that we need to unload the taxis. Imagine that we have a bunch of psychopaths in Cleveland who get into taxis and Ubers and don't leave. You don't have any taxis or Ubers for normal people, right? So the idea is not to load electrons in these molecules and just leave them there, but to load them and unload eventually. Okay? So keep it in mind when next week we talk about metabolism. I'm going to go and open the lab door for